So for today, we are doing cytoskeleton. So we have already talked about a bunch of the stuff. Last week, we kind of went over kind of the main parts of cytoskeleton, what different types are there, and then what main functions it performs. And then we had started to talk, we had talked about intermediate filaments and microtubules, and we were starting to talk about some functions of microtubules and how the microtubules are organized differently depending on the function, the ultimate function they're performing. So today we are going to continue with that. And we are going to finish talking about microtubules before going into actin filaments. And then looking at an example where different types of filaments come together to perform a very important function in our body with muscle contraction. Okay, so let's see, we were somewhere here. I know we had talked about, had just talked about the motor proteins from what I remember, right? Um, and we had started to talk about the structure. Okay, so we'll just start over here to give us a good place to start. Okay, so we're going to start off by kind of reviewing some of those microtubule functions that we talked about at the end of class last week, and then continue from there. So as we talked about, microtubules can have many different functions depending upon what cell type they are in and what type of work they need to perform, what part of cell cycle they're being a part of, or what type of just general function they're supporting of in that cell. So one of the things that they help is in neuron transmission of signals and the integrity of the neuron by creating these kind of tracts almost in the axon of the neurons that go from the neuron cell body, so nerve cell body, all the way to the axon terminal. And these tracts are kind of going in opposite direction in a way, in the sense that you have molecules that can be moving towards the axon terminal along these tracts, as well as coming back to the cell body. So they can transmit signal both towards as well as away from the axon terminal. You can also see them in our cells all along, all inside our cells. We have these microtubule structures that are providing essentially roadways for other proteins called motor proteins to move, transport other proteins, vesicles, different types of molecules from one place to another intracellularly. So this movement of these motor proteins require ATP hydrolysis. And we've talked a little bit about it last time, and we'll kind of review it soon. And they all have, even though depending on the type of cargo they're going to carry, their shape will be different. In some ways, they'll all have some type of a globular head with, amino acid residues that can interact with the microtubule directly. And then they'll usually have some types of long tail end on the other side that ends with the binding pocket for whatever cargo they are taking. Um, Again, as microtubules are part of the cytoskeletal structure, providing that scaffolding in the cell, they are also there to help position not just the proteins, but also organelles within the space so that your organelles remain in some order inside the cell structure and are not just free floating. So they're not just walking around anywhere they need to, they're not just floating in the cytoplasm. They are anchored at various places along these microtubules in specific areas of the cell. And then finally, they can be used to help cells be mobile. So different cells can have different types of ways to become mobile. They could have cilia and flagella, right? Those are usually what we think about when we think about a moving cell. And those cilium flagella contain specific types of stable microtubules. So those are microtubules that aren't polymerizing and depolymerizing. They're kind of anchored so that they are stabilized in that state, allowing the cell to use that structure for mobility functions. So first of all, going back to different motor proteins and how they move along microtubules, like I said, that you'll usually have these globular heads that have binding residues for the microtubule. They move from the minus end of the microtubule towards the plus end of the microtubule. So when I'm talking about minus versus plus, which side is getting polymerization of more molecules to create the microtubule?
The
Is it the
plus
minus end
end.
or the plus end?
The plus end.
Plus end, yes, exactly. So the plus end is where more molecules are getting added. Can somebody remind us what it is that is visually seen at the plus end of the microtubule? How do we know which side is the plus end? Oh, I know why I can't hear you guys. Sorry, I had you guys on mute. Can somebody talk now?
Would it be the GTP dimers?
Exactly. Remember how you had those GTP dimers, the microtubule little subunits with alpha and beta subunits joined together with the GTP. That's how they bind. But they are not hydrolyzed at the same rate as they bind. So you end up with this cap, what we call a cap, of these dimers, which are associated with GTP. Now, they are getting hydrolyzed though and getting converted to the GTP slowly. At some point, if that process goes faster than the new molecules that are coming in, then you start to see unraveling of that microtubule. And that's where it starts to unravel, right? But typically, the plus end is the side where you see the GTP associated with those dimers as they're coming in, and that creates a bunch of these stuck together, right? That's your plus end. Okay, so now the motor proteins don't have to carry the cargo by directly binding to it themselves, although many of them do that. And that's for two reasons, right? If each molecule that has to be transported needed a particular, basically, motor protein, right? You would need a very large variety of these motor proteins, one for each single molecule that needs to be transported. However, if you can create a kind of a general structure or a pocket or a domain to help associate these bindings, then you can have, multifunctional motor proteins that can take a bunch of different molecules or a bunch of different things based on that little domain, the binding domain being similar within them. So sometimes, yes, the cargo binds directly to the motor protein. Other times we have adapter proteins that then bind to the cargo in turn. And they allow a little bit more flexibility in what gets bound to the motor protein at the end of the day. And also allows to have larger sized molecules like big vesicles getting carried by these motor proteins without a problem. So those are, the two different ways that you can move cargo from there. Okay. So when we look at now, let's switch to a different type of function that microtubules perform, and that is movement of the actual cell, either through the help of a cilia or a flagella. In cilium and the flagellum, the microtubules are arranged in a very particular manner to create a stable structure. Now, remember, if we go back and think about cell cycle, we also used to have these anchored microtubules, right? Which were anchored along the membrane with adapter proteins. And that's what kept that cytoskeletal structure, the actual kinetochore microtubules and all the other tubules in place, right? In this case, it's a combination of anchoring as well as that structure within it. So if we were to take a cross section of a cilia or a flagella, we will see that you have this what we call a 9+2 array, where you have two microtubules, these microtubule doublets arranged in a ring structure. And these are kind of kept in place by linking proteins. So proteins that are linking those together to keep them in that structure, and by additional proteins that connect them to the central core, where there is another, in that inner sheath, there are another two microtubules, single microtubules attached to each other. So by creating the structure, it's again providing you multiple levels of stability, right? So you have those 9 doublets that are linked to each other through linking proteins. Each doublet is also linked to the central core with the help of additional proteins, right? And then within each one of these doublets, you will notice that there are 2 dynein arms. So dynein is another, right? It's one of those motor proteins. That's another one of proteins that attaches to microtubules. One of them is going to be facing towards the outer side of the microtubule. It's called the outer dynein arm. And the second one is the inner dynein arm that's towards the inside. Now, these kind of act like little oars in a way to help move the microtubule in various directions, but also to help make that structure strong and to help in that stability as well. So together with this structure, this, it almost looks like a wheel, right? If you were to look at a wheel, and that's why it is as stable as it is, because it has multiple levels of stability built into its structure. Now, what happens? How do they help us move? Well, the movement of this is dependent on those motor proteins molecules, which are the dynein molecules. And so the dynein molecules are going to move with ATP hydrolysis, in a very particular way, so that either the microtubules slide against each other or they move from one side to the other, depending on the type of movement that they need to perform. So if we were to look at just a single doublet, remember, so you have those 9 doublets along the outer side. Those doublets, the 2 microtubules that are linked in there with these dynein molecules all along, they are the ones that are going to slide up and down against each other through the movement of these dynein molecules with ATP hydrolysis. So these are the sliding movement. That's where the sliding movement is coming from. On the other hand, when you are overall looking at that structure, the entire structure of the microtubule, they not only move, just slide against each other, but they also move from side to side. So in a flagellum, what happens is that the dynein molecules will move at different rates in different parts of that microtubule from the plus to the minus end, causing it to bend to one side or the other, depending on the current or the movement of that cell at that time. And that's how those the linking proteins are able to bend. They are flexible, so they are, they are kind of like a hinge, so they can bend along with this movement, and without disrupting those doublets, they can still cause the cell to move along that structure. Then, does that make sense? Questions about its movement? So here, if you look at this is basically looking at just 2 of these doublets that are linked with the dynein arms, and it's just looking at their movement in isolation. And that's why all you're seeing is the sliding. But here, because you have all these linkers, right, in an actual flagellum, you have those rings all along the microtubule, those linking proteins are not going to let it slide off, but rather move where it can bend to one side or the other along with just a simple sliding motion. Okay?
Okay, now it makes sense. Thank you.
Okay, good. It's kind of like an engineering thing, no? Okay, so the the third function that microtubules perform is, this is a review of what we've already looked at, is the cytoskeletal structure, right? Mediated in the M phase to create those kinetochore microtubules, to create that spindle fiber structure to help in cell division. So there are two different cytoskeletal structures that mediate this process. One of them is, the main one is the microtubules, which is for the organization of the chromosomes along your metaphase plate, and then their separation to the opposing poles to create, to provide, the entire set of DNA on both sides to create your daughter cells. The second one is actually actin and myosin filaments that work in the last part of the M phase when they create that contractile ring to promote cytokinesis, to help that process of cell division. Now so we have been talking about obviously microtubules for now, but in the next part, we are going to talk also about that cytokinesis portion, where we will look at those actin and myosin filaments working together to create that contractile ring. Now, looking at that structure a little bit more in detail, again, this is a little bit of a review from Cell Cycle, which we did last week. The structure starts in the duplicated centrosomes very early in the mitosis process. So actually, this is, once the cell switches to M phase, that's one of the first things to appear, even before nuclear membrane disintegration. Those centrosomes, which have those centers of these microtubule production, will have these aster microtubules start to originate from them as they start to move away from each other to the opposite sides of the cell. And that's what's going to create that mitotic spindle. Now, if we were to look at it a little bit more in detail, we will see that these microtubules are actually quite dynamic. They are constantly, some of them are coming out and getting elongated at any given time, while others are shortening and depolymerizing as well. So if they were constantly in this dynamic form, you're not going to get a stable structure, which is what is essential for appropriate mitosis to occur. So to help that process along, there are two different ways that the microtubules are stabilized. One of the things that happens is that the interpolar microtubules, so these are towards the center of the cell, have actual interactions with each other that stabilize them so that they cannot depolymerize anymore, and they become kind of stuck in that space. While those microtubules, which are still open, which are still not bound to anything, will continue to have those that activity of depolymerization and repolymerization within them. The stabilization of these internal microtubules is essential for that structure to remain long enough and correctly formed so that you can have the metaphase spindle occur and the proper cell division happen. Now, if we were to look at it a little bit more closely, we will see that, so you have the astral microtubules. The role of the astral microtubules is to create that scaffolding around that main structure to maintain the mitotic spindle in check, to create, stabilize it by just providing it with a lot of different roads to hang on to in the cell so the cell doesn't collapse. Then you have these interpolar microtubules, right? And these are the ones that are kind of interacting with each other to create that stable structure internally within the cell. And then the third thing you have are what we call kinetochore microtubules. So the kinetochore microtubules are going to attach to the chromosome's central kinetochore, right? So these are your duplicated chromosomes. So they are two sister chromatids bound together at that kinetochore. And these microtubules are going to be bound to them. Now, they don't bind directly to them, right? They bind with the help of linker proteins again. Now, when the cell has created that metaphase plate and it is now time for them to move away from each other, a specific process occurs that triggers the sister chromatid separation. And this is done through proteolysis of the proteins that are helping these sister chromatids stay together, essentially. So there is a complex of two proteins. It's an inhibitory protein called securin that keeps this proteolytic enzyme from working unless it is needed. And it is bound to this enzyme called separase, and that enzyme is needed for this process to occur. So when this metaphase plate has been made and it is fully aligned, an active APC complex protein breaks down the inhibitory securin protein. It ubiquitinates it and targets it for proteolytic degradation. This allows the separase to become activated because it's no longer bound to securin. And that can then come in and destroy the cohesions, the associations of the kinetic of the two sister chromatids with the cohesin molecules. It can separate those, leading to their separation and movement away from the core. Now, if you were to look at this chromosome when it isn't separated, the cohesin complex, the cohesin proteins are forming kind of a ring and keeping these sister chromatids together. So when this separase breaks that down, the rings down, it essentially separates those chromatids and allows them to get moved away. Looking at it again a little bit more in detail, how it is happening within Anaphase. There are several things that are going to happen, right? So just cohesin ring separating is not going to pull the chromatids apart. It will just have them still right there in place. The second thing that happens is that the kinetochore microtubules actually starts to depolymerize. And that, as they shorten, essentially they are dragging the chromosomes along with them. So remember, you have those plus and the minus ends of the microtubules. At this point, those microtubules are essentially in that negative end because they are going to start to depolymerize. They're getting pulled apart, and the chromosomes are getting pulled apart because of that. The second thing that happens is that these microtubules, they are going to slide between each other, very similar to the ones that we saw before when we were looking at cilia and flagellar movement, right? So similar to that, the proteins linking them together are going to help them slide away as they continue to increase in length through polymerization. So in this case, you're still getting more polymerization on these interpolar microtubules. So in Anaphase, the interpolar microtubules continue to grow. Okay, so they're continuing to polymerize and continue to grow, while the kinetochore microtubules are depolymerizing and shortening. So those are two things I want you to take from here. So it's not that every single microtubule in the spindle is doing the same thing, right? The interpolar microtubules are continuing to elongate, while the kinetochore microtubules are continuing to shorten. And finally, the astral microtubules are stabilized because of their interaction with the plasma membrane linker proteins and will not be moving at all. They are the way they are to begin with. Taylor.
That's super cool.
I know, isn't it?
That's super cool.
When we learn it back in the day, ninth grade or whenever we learned this process, we are just told, okay, these spindle fibers form and then they shorten and, you know, or they pull them. Actually, they don't even tell you to shorten or elongate.
Yeah.
They pull them apart and, you know, that's it. But in reality, it's a way more complex format. And there are different workers within the same microtubule structure, right? This is working together in a very cool way. Ryan,
Yeah, that's just...
I see Ryan now. Cool. Okay. So because the interpolar microtubules are part of this internal structure of the spindle fiber and are kind of maintaining that central line, so to speak, in that cell, they actually determine where the cleavage is going to start to form. Because those bands of actin and myosin that are going to form where the interpolar microtubules had been stabilized at that point. So if you had some kind of an issue with the cell where that interpolar microtubule structure is formed a little more to the right or to the left, you will end up with those, you know, incomplete division or unequal division of cells, because you won't be able to divide it right where you need it to. So the site of cleavage furrow is going to be determined by the placement of interpolar microtubules. That's important to remember from there. Okay, so here you're just gonna look, this is just to show you a close-up of what's happening to the kinetochore microtubules. Remember, the kinetochore microtubules are the ones that are depolymerizing as and shortening as the chromosomes are getting pulled apart, right? So normally you have these ring proteins that are going around the kinetochore microtubule, and then they are linking to the chromosome to keep it attached, so to speak, with the microtubule. So the kinetochore microtubule never actually was touching the chromosome directly, but this interaction was essentially formed with the help of these linker proteins. So that means that end of the microtubule is free to do whatever it wants to do. And that's important because we needed it to have that flexibility, right? So it could continue to elongate or reduce as needed. If it was directly anchored to the chromosome, it couldn't do it. So because this end is open, it's the plus end. However, at this point, when the chromosomes are all lined up and the cohesin ring is broken down, these are going to have, are you laughing at me, Taylor?
No, I was laughing at Ryan's comment. It is so true.
Looks like a pencil sharpener. It does. It does kind of look like a pencil sharpener.
It took me off for a second.
No problem. I was like, what did I say? But yes, because that end is open, and remember, these are all going to be the ones that are associated with GTP, right? They are not yet hydrolyzed. So these are going to rapidly hydrolyze. And because that's going to be such a rapid hydrolysis of these molecules, they're going to start essentially ripping off that tube. And so all these tubulin units, subunits, which are still in their dimer forms, they're alpha and beta together, but now it's a GDP instead of GDP attached to them, will start to pull off. And so this is going to start to become shortened. So as they shorten, this linkage is still there, right? Those proteins are still, the ring proteins are still there. The anchor to the kinetochore plate is still there. It kind of looks like a hot air balloon, like the basket of the hot air balloon to me. But the way they've drawn it a little bit differently than what I'm used to usually. But the chromosome then just kind of travels along with it and it goes to the other end. There is kind of like an animated video of this in this link. I'll see if it works. I remember that was a cute one. Do it. Doesn't like me. Maybe it will. We'll see. So just reviewing microtubules. Microtubules are different than the intermediate filaments in that intermediate filaments were those long kind of ropey things that were coiled along each other, right? You had them coiled as dimers, then coiled as tetramers and octamers, and then that's what was making a very thick rope essentially. And you could line them up next to each other and get long, strong ropes. This one are hollow tubes. And they have very different ends, structurally distinct ends, which make sure that you know a plus versus a minus end. A plus end is the side is getting more and more dimers getting put in, the tubulin dimers coming in, elongating the microtubule. And the minus end is the side that is not having any additional monomers or dimers getting put in. The centrosome is the major organizing center where these microtubules originate in animal cells. And so they're dynamic. In general, they are going to have instability because they're constantly going to be some balance between adding more dimers and removing dimers. So they're polymerizing and depolymerizing all the time, unless it is stabilized through interaction with linker proteins of various types as anchor points to control that dynamic movement. In this dynamic instability, this whole process of polymerization and depolymerization is driven by GDP hydrolysis, and it can be modified by use of certain drugs that can either stabilize them or and cause them to freeze in that way, or it could ever prevent them from creating them to begin with. They help organize the cell interior. They keep all the proteins in place where they need to be. They keep all the organelles in place where they need to be. They drive intracellular transport by allowing microtubules to have these racetracks essentially to use for movement of molecules and cargo and vesicles across from one place in the cell to another. And they also help the cell move by creating a very stable structure in the form of cilia and flagella. Finally, they can also help you with cell division. Now, in cilia and flagella, the microtubules are stable and not dynamic, and they are moved with the help of motor proteins, specifically called dynein. Okay, so I'm going to stop the share for a second and see if we can look at that link, because it was a good link. Why won't it let us show you? Share screen. In the meantime, do you guys have any questions?
I had a question about that ring, the one that pulling the chromosome. Is that one like moving along with?
Yes, so it is a very strong binding to the chromosome on that one. And the ring itself is just like a ring around. It's not having direct interaction with the kinetochore. It's around it. It's kind of, placed around it, but it kind of moves along with it because it doesn't dissociate from it. Does that make sense?
So how it's moving with the microtubule when the microtubule gets hydrolyzing, so it cannot bind to it, it's going the other side?
That's what I said, that it's not directly binding to the kinetochore. It's kind of creating this ring structure around it to keep it in place. It's inserted through it. The microtubules is inserted through it. So it gets pushed along with it because of that.
Oh, okay.
It's like, when we have, I don't know, those napkin rings. So if we were to push something through it, the, napkin being microtubule, it's a very bad example of a microtubule, but that's kind of the way it is. Okay, where is that thing? Right there. Let's see if it will do it now. We're not going to watch the full 9 minutes, by the way.
Machines that, but through science, we do have a
There's
fairly
a
good,
little
as part of their
animation
thinking is the moon.
inside
And
it.
he
I have to know.
transformed our understanding of the moon. The perception of the 17th century was the moon was a perfect heavenly sphere. But what Galileo saw was a rocky, barren world, which he, through his watercolor paintings, another scientist with very big ideas.
Towards the end, they talk a lot about the actual, you know,
that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have two strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it right here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes. We have no idea, really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently.
So you see how like they are all along it, right? And they're forming this structure. So you have these one set of linker proteins that are kind of hanging on to the microtubule, but then you have all these kind of these thread linker proteins coming off of it that are then interacting with other proteins on the other side of the chromosome.
It's involved with the growing of the microtubules.
And so here you see the growing of the microtubules happening. And then
it's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier.
That's the part that I wanted to show you. Osana, did that answer your question better than me?
It does, actually.
I know. That's why I wanted to show you. Because I can't really show you unless I like stand at a thing, drawing it out for you, or showing it to you like this in an animation. Isn't that the coolest?
No way, that's super cool looking.
I know, and that's inside your cells all the time. That's exactly what they look like, actually, in real life. So
no way.
Yeah. That's what he was telling you too, that this is literally derived exactly from science. That's the way they look inside your body.
That's so weird.
Not in those colors, but otherwise.
Yeah.
Yeah.
That's so weird.
That's the way they look. It's super cool. Okay. Going back to our lecture, boring lecture again. Yeah, Ryan likes it too. It is wow. I love motor proteins. They're so cool. Okay, so moving on to actin. So that's the third of the cytoskeletal structures that we're going to talk about. Now, actin filaments actually work very similar to microtubules in the sense that they allow the cell to maintain its structure and its shape in the way in the cell in the way that it needs to perform its function. So that's a big part of what it does. But it also has other types of functions as well, one of which we already kind of know about. That was in that contractile ring in cytokinesis. It also can help with movement of the cell in a different way, and we'll talk about those as well. It can help the cell sense and respond to signals around it. through these anchor points all along the cell that are maintaining its shape and its kind of space in life in the cell, in the wherever environment they are in. So actin filaments are different than microtubules in that they are a lot thinner and they are not hollow like, again, microtubules were. These are filaments, they're thin, they're flexible protein threads. They almost in structure remind you of intermediate filaments, actually. So if you think about that, their final form reminds you of that, but it is different. They are also made very similar to microtubules with these dimers coming together, polymerization and depolymerization. Um, in this case, however, you just have actin monomers that are creating that structure. So you don't have an alpha and a beta subunit, but just a single actin monomer looks like these two globular heads that then stick together to create a rope-like structure that then coils around each other to create a, coiled thread, essentially a coiled thread filament. They will also have a plus and a minus end. These are a lot thinner than your microtubule structures. Um, and they are clearly visible inside our cells. So an actin monomer works, like I said, very similar to tubular polymers, tubulin, and the way they polymerize. They both have in a positive and a minus end. And the actin monomer works, however, through ATP hydrolysis instead of GTP hydrolysis. In this one, what you will notice is that the actin gets bound to ATP to activate it, which then gets pulled into the plus end of the actin fiber, where as it becomes dephosphorylated, right, as it becomes hydrolyzed, the ADP is released and the actin structure will move towards the minus end. The way it is working, you see these 1, 2, 3 sites because at any given time, generally in this case, you will have only 3 molecules of ATP associated actin monomers attached in that actin filament. It won't be as extensive as you see in a microtubule, right? And again, they kind of work the same way, that the dynamic instability of both actin and microtubule work similarly, where there are constantly some balance between dimers coming in or monomers coming in this case, or and coming off of that. So some of the things that we need to now look at is, well, how do they function? So they actually function with the help of binding partners. And depending on which binding partner is associating with the actin filaments, very different functions are going to be performed at the end of the day. So the monomers are actually not able to just go directly to the actin filaments. They require binding partner in the form of monomer sequestering proteins that are bound to these monomers at all times, unless a process causes them to be dissociated with this sequestering protein. So when they're sequestered with the binding to this protein, they're not going to be able to attach here, right? They won't be available to be added on to an actin filament. Different processes, different signals cause this process to be dissociated. Again, there's some type of basal level of instability in there. So there's always some molecules that are free and some that are bound. And depending on the environmental stimulus or the stimulus inside the cell, this actin monomer that are freely available to create filaments is going to change over time. Once they are formed into the filaments themselves are also anchored, usually using other proteins that are going to be part of either maintaining that structure or moving it in a way that it needs to get to the right place. So some of the proteins that are known for helping it do its function right? So these are different behaviors that you'll see in actin filaments in our normal cells, in animal cells especially. So they can obviously form just scaffolding in our cells, right? That's one of its main function, is to create the scaffolding structure in the inner membrane of the cell, right? In the inside of the cell, in what we call cell cortex. And that scaffolding is maintained in its structure with the help of these cross-linking proteins. So if any of you have done like tile work, you know how we add those little spacers in between? They almost remind you of that. They're like these little linkers that keep those in place in there. They can also help, they can also be stabilized. So again, they're going to be just like myotubules normally with dynamic instability, constantly polymerizing, depolymerizing, but they can be capped off on the plus end with a capping protein, which then stops it from elongating further. They can still depolymerize and shorten, but they cannot elongate any further. So in certain cases, you can block the plus end so that they no longer polymerize anymore and no longer continue to grow. They can also have proteins along its sides to create a stable structure that will be part of a tissue or part of an organ system. This is how our animal fibers especially work actually, is with the help of these binding partners. So tropomyosin is one of those proteins that can wrap around the filament and maintain its structure and position and kind of stabilize it. And then myosin motor protein is a set of proteins that can help move these individual filaments, again, sliding them along each other. A little bit similar to dynein, except the myosin motor protein is interacting with adjacent filaments. It's not, both of its globular heads are not on the same side of the, or on the same actin filament, right? So with the dynein, you had the filaments with the globular heads along one side. With this one, the myosin motor protein, the globular head, they're laying sideways instead of standing up. You can also increase the strength of the actin filaments structure by joining them together. Now, in intermediate filaments, they were all coiled against each other, right? They were just kind of binding and coiling around. In this case, you actually don't do that. You just have those dimers that are there, right? You have the initial coiled filament, but those coiled filaments are linked together with what we call bundling protein. This is something you'll see in Filopodia or other kind of pseudopodia structures to help in stabilizing that structure and help in movement of that cell. And then finally, you have these proteins called severing proteins that can break off pieces of this actin filament to create smaller pieces for specific functions inside the cell as well. So again, just like with microtubules and just like with intermediate filaments, you can inhibit actin function and actin production and the way they are made using specific drugs. 3 common drugs that are known for affecting actin fibers directly are phalloidin, cytoskeleton, and electrochlorin. Now, phalloidin is something that I would want you to know about, and you should be able to kind of compare it with Taxol, which was affecting microtubules. So phalloidin is, again, a drug that stabilizes filaments, very similar to Taxol. But in this case, this is binding to actin, and Taxol is binding to the microtubules. Okay? Cytoskeleton is a capping protein. Essentially, it will cap the filament's plus end and prevent polymerization so they can no longer elongate. And letrunculin is a binding binds the individual monomers, so it's a sequestering protein, so it won't let the filaments ever form. Okay. So if you're told what it does, you should be able to, on the test or a quiz, you should be able to categorize it as a sequestering protein or a cross-linking protein or a severing protein, and so on, okay? Okay, so let's look at some of those areas within, how are we doing on time? Oh, we're good. So let's look at some of those areas that actin is very active and that it's working in. So the first one that we're going to look at is cortex, which is a major component, which major function associated with actin filaments. So the cortex is essentially a scaffolding, right? That is all along the inside of the cell, maintaining its shape, maintaining its integrity in space. The cortex essentially is a bunch of these actin filaments connected together with the help of attachment proteins that link it to the membrane directly. And then the actin filaments themselves are also linked together with the help of attachment proteins as well. They are also connected, associated with these spectrin dimers, which are small kind of, those ties that we get, those little, they come in many things nowadays. All the toys or anything you buy from the store, they have these little ties attached in the back. They're impossible to get off, all of them from the toys. That's what they are. So spectrin dimers are essentially helping to anchor and join these actin-binding filaments together. So if you look at this, all these red are those little ties that are connecting actin filaments to each other. And then, so you just see a little tiny piece of the actin. It would be all over the place. They would be creating that bigger structure of the scaffolding. And then the spectrin dimers would be those thin filaments joining all that scaffolding together. And then these globular proteins that you see are connecting the anchorage points of these filaments to the plasma membrane. So that's how they usually work. The attachment proteins usually interact with transmembrane proteins directly and not with the lipid portion of the plasma membrane. Again, there's a little video, Life of a Cell, that I really like. You guys should watch it. It's fun. We may watch it next time as well. Now, one of the things that actin is known for is to help move the cell, any cell, move in its environment. So even these cells, the cells that you looked at under the microscope in our labs, those cells, they are adhered to the surface, but they are still mobile. They are still moving. And so if we were to look at them under a time-lapse microscopy, we see that they move in space over time. And they do, some of them just kind of wiggle in a small space. Some of them are very motile and can move even long distances over a very short period of time. All those movements are actually happening because of this actin cortex and the way these actin molecules present in that cortex are polymerizing or moving in that space. So the actin cortex, if the cell was in a completely stable space where it was not going to move at all, the actin cortex would actually be evenly distributed all throughout the cell and would overall be in a balanced state where all areas are going to have the same amount of polymerization and depolymerization. However, if there is some kind of a signal that is helping the cell move in a particular direction, it causes kind of a tension or usually it's done through ionic channels, ion channels. It causes an influx of ions towards one side, causing these lamellipodium to form, which then cause that side of the actin fibers to become what we would have as plus end. So the plus end is what protruding into this lamellipodium as more actin monomers are getting attached. It's polymerizing, extending, and elongating. And that is how it's going to slowly move towards one side or the other. Another thing that helps in this movement is the help of myosin motor proteins that help those actin 5 filaments that are inside it slide against each other and move in the direction of the polymerization. So it will continue to do that until the signal stops. Until then, it would continue to move. Now, this is a slower movement than what you would see with a cellular flagella, right? It's a much slower process. It's not going to be a cell just kind of skittering away. It will be over a longer period of time, a slow movement in a particular direction. And this can happen in just a single direction, or the cell may be getting signals from multiple sides, in which case it may just kind of move a little bit as in wiggle and not just move in any given space. Now, how these protrusions form is, again, like I said, dependent on those actin monomers getting attached and polymerizing those actin filaments that are part of the cortex. So if you were to look at it in a close way, so this is an electron micrograph of the actin filaments in that cortex space. So it's not a very, just a sparse structure. It's a very, very dense structure. It's like a thorny bush, right? Looking at it closely, you see your different actin monomers. These are your actin filaments and the anchor proteins that are keeping them together. Some of them are capped, so they cannot polymerize. But in the front end, the leading edge of the cell, the side of the cell that had that ion influx, inducing this movement, you will see a lot of polymerization happening. That's what's your leading edge of the cell. That's where you're going to start to see the lamellipodium happening. On the opposite side of the cell, you will see the opposite happening. Those are going to depolymerize as they get shifted forward, because the membrane potential has shifted to the front end in a way that that's become the leading edge. So in the lagging side of the cell, you will see depolymerization of actin monomers. And on the leading side of the cell, you will see polymerization of the actin filaments. And so what controls that is by far extracellular signals, right? It is because the cell received a signal that induced either a membrane potential or ion channel opening, causing one side of the cell, one part of the cell to become polymerized or to become that leading edge. So if you have a completely unstimulated cell, a cell that is normally in its space the way it needs to be, what you will see is a cell that is evenly distributed, its cytoplasm, it's all in its same place. And the cytoskeletal structure is well maintained throughout the cell. You can have different types of signaling cascades activated to give very different results. Now in row activation, you see that typical leading edge of the cell form, where you are now going to see lamellipodium form towards that leading edge, and the cell is going to start to move, in this case, over here in the, can you see my arrow, by the way? Right?
Yes, we can see it.
So, yeah, so it's gonna basically move down, because this is where all the new structures are forming, all the new filaments are forming. And the backside is where they're depolymerizing. And you can see that because these membranes are kind of ripping off almost from the surface of the cell. If you have CDC42 activation on the other end, that is a very different type of activation, which is kind of causing the cell to lift up and ball up essentially, right? So in this case, the cortex is actually depolymerizing all along the cortex and not just in one place. So these, depending on what type of signal you're receiving, the microtubules as well as the actin filaments are going to respond in a very different way. And in this case, specifically, the actin filaments are the ones that are most prone to responding to these extracellular signal, because that's the first line of defense or the first line of structure inside the cell in the presence of the cortex, in the shape of the cortex. Okay, so now going back to, again, the cell cycle and looking at the animal cells and the way they divide. Once your microtubules have separated the chromosomes into their opposing poles, you have those interpolar, microtubules that are going to dictate where this contractile ring is going to start to form. Now, this is made up of actin and myosin filaments joined in, again, the same way where it is going to constrict as other side by side with motor proteins in between to move them along so that they can move, constrict that space until cytokinesis happens. So actin filaments are thin and flexible, right? And they polymerize and depolymerize very similar to tubulin, except in this case, it is run through ATP hydrolysis. And you have actin monomers that are coming together instead of tubulin dimers, which are composed of two types, right? Alpha and beta. Actin functions with the help of binding proteins, and the binding partner that it is bound to is going to dictate what function it will perform in that space. So they completely change their properties based on what they're binding to. The biggest structure of actin that we look at that is constantly responding to stimuli from outside is the cortex, which is extremely rich in actin filaments and create the scaffolding of the cell. And it helps not only the cell maintain its structure, but also respond to environmental stimuli by helping it move in respond to it. And they do that by using the help of actin binding proteins that help protrusions form at the leading edge of the cell and move the cell in the direction of the leading edge. The actin can also associate with myosin and form contractile or constricting structures, as we see in cytokinesis and also in muscle contractions. So finally, we are going to talk about muscles. So muscles are actually pretty cool because their whole function is to contract and relax and to help us as organisms move in space in response to stimuli in our environment. So what happens in a tiny cell, right, in a small cell internally with the help of actin, myosin, microtubules, and intermediate filaments is now happening at a much grander scale in the form of these muscles, right, at an organismal scale. So in these contractile structures in the muscle, you again have actin and myosin working together to form contractile structures. If you look at individual myosin molecules, or the monomers essentially of myosin, they are composed of globular heads, like you've seen, with many motor proteins, along with a long tail end that can bind to each other, and the heads are what are going to interact with the actin filaments to help it move. So these myosin molecules interact with each other through the tails. The heads are then free to associate with their binding partners, in this case with actin filaments. And they do that by interacting with the minus end of the actin filaments, leaving the plus end free to the polymerization and to increase and shorten as needed. So if we were to look at a myosin filament, what you will see is that on both sides of that filament, you will have essentially lots and lots of these heads sticking out. And in the central space, there'll be a little gap where the main tails of both ends are combined together. Each of these myosin filaments is usually a micrometer in length. So inside our muscles main core is made of these sarcomeres, which are individual kind of unit of muscle fibers, or the contractile unit of muscle fibers. In this case, what you have are bundles of myofibrils together in a structure to create a big disc And so a skeletal muscle cell, these are like your myotubes that you were going to see, right? That we were trying to generate. It's the same idea. It's basically a muscle cell that's packed with these myofibrils that are composed of these actin and the myosins together. If you were to look at a cross section of it, you will see two large myofibrils lined up next to each other with overlapping region in the center where you see those myosin filaments sliding against each other. A sarcomere, or one portion of this one contractile unit, is approximately 2.5 micrometer in length. So this central area is where you have those myosin filaments with their tails, which is taking about half of that space, with the additional space on either side taken by the actin filaments. So all along the side, you just have actin filaments with no, and those would be your plus end of the actin filaments. Inside, you have the actin and myosin bound together to help it slide against each other to move. So let's look at that a little bit more in detail. These are anchored at these Z discs on either side of a sarcomere. And inside you have essentially the plus end of your actin filament and the minus end associating with the or plus end of the actin filament with the minus end associated with the myosin filaments. In a typical position, when the cells are in a completely relaxed manner, these filaments, the myosin filaments, are largely open and not bound to any actin molecule or actin filament. Just on the edges, there is just a little bit of interaction. However, when they're fully contracted, all the myosin heads are going to be interacting with actin filaments, and only the tails are going to be exposed. Everything else, so it's going to be a marked shortening of that sarcomere when it is in fully contracted space. So this movement is done very similar to how other motor proteins move, right? It's the same idea at the end of the day. It is done through ATP hydrolysis. So myosin, both the polymerization and depolymerization of myosin, as well as its movement with interaction to, actin polymerization and depolymerization, as well as its interaction with myosin, are both requiring ATP, right? So all of this is through ATP hydrolysis. And the movement is dependent on these myosin, the globular heads of the myosin, binding to the actin molecules and moving them from one position to the other. And they do that by taking a myosin filament, which is fully loaded with ATP. And as it gets dephosphorylated, it binds to the actin molecule in that dephosphorylated state and pushes it forward with the help of that dephosphorylation energy. And then it releases it in the next round of ATP hydrolysis. So starting at the beginning, the myosin head is attached to the actin filament. It is released when the ATP is bound to it. When it's hydrolyzed, it goes back and binds to the next position on the actin filament, which causes it to get moved essentially. And then it will be back in its attached space once ADP is released until the next ATP molecule comes in. Okay, questions about this? Good. Sounds good. What causes this is, again, a lot of actin response, you will notice, will be through ion channels, whether it is at the lamellipodia or it is, or any type of movement related function, or if it is in muscle contraction. It's the same idea, but those ions change with the environment where they are, whether it's a sodium potassium ion channel versus whether it is a calcium concentration channel. So a muscle contraction happens due to a sudden rise in cytosolic calcium concentration. And it's usually done because there was some type of voltage gated channel that was activated in response to an action potential, leading to an influx of calcium to be released into the cytosolic space, causing the myofibril contraction. That's what triggers those system to work. Another protein that controls how much a muscle can contract or relax is tropomyosin. It's another binding partner of actin. And tropomyosin works with additional proteins called troponin complexes. So these are multiple proteins that come together to create kind of linker proteins between tropomyosin and actin molecules. The tropomyosin blocks myosin's binding sites so that it cannot be activated and it cannot bind appropriately to an actin filament or detach from actin filaments. However, when there's that influx of calcium, the tropomyosin can no longer bind to that site and makes it open. So now the binding sites on actins for myosin are open for attachment and can cause it to be attached and be stabilized and move along with it. So normally, when there is a completely relaxed state and you are not having that interaction, the tropomyosin is what's blocking that myosin binding site on actin molecules. Okay? So muscle contraction depends on the interacting filaments of actin and myosin. Myosin is essentially a motor protein that combines together in these long filamentous form. There are a bunch of these together, bound together through interactions with their tails. Actin filaments slide against the myosin filaments during muscle contraction through interactions directly with the myosin binding heads. Yeah, I think we are done actually almost. I think this is the last slide. And that muscle contraction is triggered again by a sudden rise in cytosolic calcium ions in this case. And obviously different muscles will perform different functions depending on where they are. So with that, probably. Well, you can always go back and listen to it again and take more notes. So we're going to stop here. And next week, or not next week, on Thursday, we are going to start learning about cell communities and focusing on how cells communicate with each other. Sounds good?
Yes, thank you.
Welcome. Take care.
I only have